Hello, everyone. Thank you so much for coming out to hear my talk today. I apologize in advance that I could not do this live, but unfortunately I'm traveling during this time. But it's really my pleasure to take this opportunity to talk to all of you about some of the research that we have going on in our lab that might be relevant to the urobiome and the experimental evolutionary approaches that we're currently taking. So my name is Megan Behringer, and I'm an assistant professor at Vanderbilt University. In particular, my lab is interested in eco-evolutionary feedbacks. In particular, how does the environment influence microbial evolution? And then how does microbial evolution alter the environment? And we can think of this as a cycle because imagine an environment that is particularly stressful. Maybe that induces a mutation in a microbe in that population. That mutation happens to be beneficial, natural selection promotes it, but that mutation also might result in some type of metabolic byproduct or some type of behavior by the bacteria that might be aggressive towards other bacteria. And now that evolution of that microbe has changed the environment and the conditions that it's in. If it's a metabolic byproduct, that metabolic byproduct could be toxic to the community. And so now they must not just adapt to the environment which they were in before, but they must now evolve new adaptations in order to bypass that toxic byproduct. And so you can imagine here how this can be quite cyclical. And to investigate this, we use an approach called experimental evolution. So in my portion of the talk, I'm going to be discussing how the environment influences non-adaptive microbial evolution, while I'm going to leave the more adaptive microbial evolution to Owen in his portion of the talk, where he's going to discuss some really exciting, not yet published results that are coming out of his dissertation research. Now, to begin to talk about non-adaptive evolution, it's important for me to first introduce the 5 forces of evolution: mutation, recombination, migration, random genetic drift, and natural selection. Now, these 4 blue forces we think of as non-adaptive evolutionary forces, which means they occur non-deterministically and at random, while natural selection is the only adaptive evolutionary force that we think of in evolution. Now, again, the force that I'm going to mainly focus on in this talk is mutation, and we're going to normally investigate that through the lens of experimental evolution using an approach called mutation accumulation. And so to do that, we need to make the effective population size very, very small in order to minimize as much as possible that effect of natural selection, so that we can actually get an idea of the true mutation rates independent of if these mutations are beneficial or deleterious or neutral. And so we do this by introducing a single cell bottleneck at each transfer. So as you can see in this figure here, from this really nice paper by Michael McDonald that was in EMBO Reports, we streak out the bacteria on a plate, aiming for a population of single colonies that are isolated. You pick at random a single isolated colony. We usually do this by dotting the back of the plate and picking whichever isolated colony is closest to the dot. Once we do that, we streak it, right? So that we hope that every new colony now has arisen from a single cell. We know that's not always particularly true. But in doing that, we also know that there's about 20 generations per plate. And then if we look at this process through time, we get an effective population size of about 10. So really, really minimizing the strength of selection. Something would have to have a selection rate of 0.1, which is an extremely large selection rate in order for natural selection to bias this project. And so we're really able to get an idea of what spontaneous mutation looks like. And so some motivating data we had for our mutation experimental evolution studies kind of came from this one paper that we read in PLOS Genetics that discussed this anaerobically grown Escherichia coli had an enhanced mutation rate and a distinct mutational spectrum. And here the authors found that when they were working with E. coli B and they were cultivating the bacteria under anaerobic or an aerobic environment, that they were seeing higher mutation rates in an anaerobic environment, if not kind of statistically similar mutation rates in an anaerobic environment as they do in an aerobic environment. And to me, I found that kind of surprising. And so one of the first orders that I had as I started my new lab is that we kind of deputized a really talented former undergraduate in our lab, Michelle Yin, who is now in medical school at Tulane, to kind of investigate this a little further. And so we used at first this kind of quick and dirty approach called fluctuation tests. These are based on reporter loci. In this case, our reporter loci is RPOB. And certain mutations in RPOB can confer rifampicin resistance. And so essentially what we do is we culture about 20 tube cultures of bacteria, and then we plate each one of these cultures onto individual plates containing rifamycin. All the mutations that arose would have arose in that single overnight culture, and so we know how many generations they have, how many cells they are, and then we can look at the variation across the plates in which mutations, rifamycin resistant mutations occur. We know how many mutations in the RPOB locus can confer rifamycin resistance. And from this way, we can kind of get a really rough per genome count of mutation rates. And so we repeated this experiment in our lab's focus E. coli strain, which is E. coli K12MG1655. And repeated this on Davis Mineral Media. And what we saw was, again, kind of a statistically insignificant difference between aerobic and anaerobic mutation rates. However, in our case, we saw that the aerobic mutation rate might be slightly higher. And so we wanted to see actually how this played across different media. And so Michelle did kind of a large swath of the different medias that we have in our lab. M9 plus 0.2% glucose, LB Miller, Easy Rich with 0.2% glucose, NYC3, and synthetic human urine. And across the board, we were seeing that the mutation rates were slightly higher and sometimes significantly higher, always in the aerobic condition. So we plotted these results that Michelle had, and we found something kind of surprising, and it's that the mutation rate differences not only varied between oxygen conditions, but this was greatly influenced by the primary carbon sources. So in things like easy rich, LB-based medias, these are all very heavy on the amino acids and despite them containing glucose, the amino acids are really the primary carbon source that you're seeing there overwhelmingly. And in these cases, we all saw the mutation rates being much higher in aerobic conditions and lower in the anaerobic conditions. However, when glucose was the primary carbon source, we saw that the mutation rates were quite comparable between the aerobic and anaerobic conditions. We were trying to figure out why this might be, and one of this explanations that kind of came to us was the different metabolisms that we're seeing when you're metabolizing primarily amino acids versus glucose. And that's when E. coli are metabolizing glucose, it's very easy for their metabolisms to get overwhelmed. They end up in overflow metabolism, which essentially they're now fermenting whether or not they're aerobic and anaerobic. And because they're doing that same fermentation metabolism, both aerobically and anaerobically, we're seeing that that type of metabolism they're doing is likely having an overwhelming effect on their mutation rate, not just the kind of environmental oxygen conditions, but the metabolism that they're doing because of their environment. And so that kind of made us think a little bit about what was going on across the bacterial tree of life, and that we know that mutation rates vary in bacteria, but most of these mutation rates that we've kind of looked across this bacterial tree of life, while very exhaustive sequencing, you can see we've got most of the major phyla covered, and a lot of the model organisms covered. All of these species we're seeing that have been previously assessed are aerobic. Now, this is a major problem, and one of the reasons is that if we think back to the evolution of Earth, right? And what oxygen looked like in early Earth when bacteria initially evolved, right? We were in an anaerobic Earth, and it's not until maybe what 2.25 billion years ago that we even transitioned to an oxygenated atmosphere. And so all of the life on Earth is built on this chassis of anaerobic respiration. And so understanding what anaerobic mutation rates look like is very, very important. But also we can think in terms of biomedicine, right? We know that the niches across the GI tract or even the urinary tract vary greatly in the amount of oxygen that is there. And this all is going to kind of, as well as the resources, and this is all going to contribute to what these mutation rates look like. And if we want to understand this microbial evolution, we want to look at it through genomic sequencing data, longitudinal data, or even just kind of isolates that we're going to build phylogenies from, and we want to infer molecular evolution rates and kind of put that onto time, we really need to understand what the base genetic spontaneous mutation rate looks like in order to apply that molecular clock. And so in this effort, I'm going to present some of our recently published research. This is actually in the December issue of MBio with Owen as the first author, titled Elevated Rates and Biospectra of Mutations in Anaerobically Cultured Lactic Acid Bacteria. And this is where we're going to start to get into our urobiome relevance. And so we chose three species of lactic acid bacteria. We chose Lactobacillus crispatus, clearly because it's for its urorelevance, but then we also chose a very closely related Lactobacillus acidophilus, in order to kind of look at how much of this molecular phenotype that we're going to look at in mutation rate might be specific to the clade. And then we also picked an outgroup lactic acid bacteria, Lactococcus lactis. We also picked L. crispatus is very interesting because it differs from L. acidophilus in just the presence of different aerobic metabolism and electron transport chain genes, as well as genes that are important for oxygen radical scavenging and other antioxidant systems. And the interesting result that we saw here, really first off the bat, is that we saw extremely high mutation rates in Lactobacillus acidophilus. Now, mind you, this experiment was conducted completely anaerobically in an anaerobic chamber. The cells were incubated. We have an incubator within the anaerobic chamber. And so they only left at the very end of the experiment. And even we extracted the DNA initially within the anaerobic chamber. And so all these high mutation results are all specific to that broadly anaerobic environment. And we're seeing almost one of the almost highest mutation rates that we know in a cellular organism coming out in Lactobacillus acidophilus, with Lactobacillus crispatus and Lactococcus lactis kind of lagging behind, having very similar mutation rates. And this dotted line we included here is actually, we found another experiment that had looked at Lactococcus lactis previously, but aerobically. And we see that that aerobic mutation rate is slightly elevated compared to the anaerobic mutation rate, which is consistent with the idea that, aerobic respiration should induce, more oxygen radicals and increase that mutation rate. But still surprising that in this anaerobic environment, we're seeing such high mutation rates. I also want to point out exactly where aerobic E. coli is to kind of give a little bit more context into how elevated these mutation rates are and just exactly how special lactic acid bacteria seem to be right now in the context of its mutation rate. And so we can also look at these mutation rate spectrums because we've done whole genome sequencing on these mutation accumulation lines. And we see that the high mutation rate, while visible in many of the different single nucleotide mutation types, for all three species, we see the highest mutation rates happening in the GC to AT T mutations. Um, because lactobacillus had such a high mutation rate, Owen was really kind of interested to see if there was any kind of positional bias, as positional bias had been kind of seen before looking in different bacteria with high mutation rates, particularly mismatch repair resistant Vibrio species under mutation accumulation. And interesting, unlike the Vibrio species, where the positional bias was kind of more towards the middle of the replication trajectory, we're seeing that Lactobacillus acidophilus has a very high positional bias near the ori. And so you can kind of see that here, particularly even in those GC to AT mutations where this is darker, a really high mutation rate by the ori, which tapers off on both sides of the replication port as we get closer to the ter. We can plot that kind of again to kind of show you exactly like how biased this is, and you can see that it is very like uniquely elevated when you plot it this way, and particularly around those GC to AT mutations in acidophilus. So we wanted to kind of understand where this was coming from. As I said before, this had only kind of been seen in Vibrio, and particularly a mismatch repair deficient strain of Vibrio. Um, and so Owen kind of went deep to do a comparative genomic analysis across all the Lactobacillus acidophilus genomes that he could find in NCBI. And one of the new things that we found is that this enrichment for either loss or mutation of mismatch repair proteins that is widespread across the Lactobacillus acidophilus species. While these couple of wild strains seem to be mainly intact, as we get into the other strains, particularly the type strains and then the commercial strains, right? We're seeing kind of that widespread loss of different DNA repair proteins, which could be associated with this domestication status. We can see a little bit more specific about where these are showing up, but particularly mut-S is mutated in across the type strains and in some of the commercial strains, which is going to contribute to a very, very high mutation rate. In E. coli, it's about 100 to 150x increase in mutation rate, but as well as some of these other DNA repair genes. And so this could be a warning for future probiotic development that as we kind of domesticate these strains, as they've gone through massive bottlenecks, that you can have these unwanted mutations fixed. And this could be important if we're trying to kind of maintain the genetic integrity of the strains. If you want consistency in batches of probiotics, if you want consistency across your experiments, right? The fact that these bacteria have such a high mutation rate, we really have to think about like how we're maintaining the lines, how we're maintaining the stocks. I certainly would cringe at this point at anybody who's keeping just large swaths of Lactobacillus acidophilus on like a slant somewhere, because those have certainly accumulated a number of mutations while sitting on that slant. We also kind of went forward to see what other kind of mutations that we were seeing across the lactic acid bacteria. And we see that Lactobacillus crispatus exhibits uniquely high structural variation and uniquely high rates of structural variation, particularly in large deletions or in transposition. And this kind of manifests also, we saw in a unique way within the CRISPR loci, where we were seeing different deletions of spacer regions within the CRISPR loci, which is kind of unique because we typically think of CRISPR loci as they evolved as you gain and lose parts of the CRISPR loci, that the spacer and the repeat should all go as one package, but we're seeing just the spacer go, but the repeat being maintained. Um, and so what we might be looking here is maybe some insight into actually like the processes and steps of how CRISPR loci actually evolve spontaneously. But kind of something to look out for and kind of something unique about Lactobacillus crispatus because the gain and loss of spontaneous mutations in CRISPR loci hadn't really been reported yet. But LB crispatus, due to this high rate across the 96 MA lines might be a really unique model species for looking at the evolution of CRISPR loci, especially in that spontaneous mutation context. And so the summary I kind of want to tell about the mutations in lactic acid bacteria is that mutation rates are heavily influenced by diet, environment, and demography. That lactic acid bacteria have uniquely elevated mutation rates. This can contribute to rapid evolution, but is concerning in the context of genetic load. Domestication and industrialization of species can result in increased mutation rates. And that Lactobacillus crispatus in particular exhibits elevated structural variation rates and spontaneous mutations of the CRISPR locus, which could inspire even questions of what the urovirome looks like, what the phages look like in the urinary microbiome, and how they might be influencing the microbial population that is there, and how long that some of these lactic acid bacteria are able to kind of maintain those CRISPR loci to kind of balance out their population with the phages that are present. So now I would like to take an opportunity to introduce Owen's work to understand how the environment influences microbial evolution. But before I do that, I want to introduce how I personally think of infection as an evolutionary biologist and a microbial ecologist. And I've recently become really inspired by stream ecology, so that kind of might explain some of this metaphor. But really, I like to think of infection as an invasive species question. So if you look at this conspicuously shaped watershed as an example, where we have a river, right, that flows, and in its mouth here into a larger water source is very close to this lake here. And between the river and lake, we have a confluence, right? And now this river will have its own biological and physical properties based on pH, flow rates, biological makeup, the community members that are in it, the metabolites that are present, and similarly, the lake could differ. Um, and the biota that are in here, the ecology of the river and the lake can be quite healthy. Um, but if something happens, maybe physically to the confluence, that now all of a sudden microbes from the river start flowing into the lake, right? Some of these individuals can really start to disrupt the ecology, really in a way that is detrimental to the health of the lake. And now we're starting to have an invasive species problem. And I think of this in analogy, and I kind of really like it because it reminds me of a visual that I've seen in a really nice review paper from Scott Holgren's group, where we kind of see the gastrointestinal tract and the urinary tract and the sharing of microbes between these two biological watersheds, for lack of a better description. Um, and Ashley Ellis had a really nice paper on this too, kind of actually individually tracking the bacterial strains that we're seeing in the gut as they make their way through and eventually manifesting um, in the urine or as a diagnosed urinary tract infection. And so in this case, you can almost think of E. coli in this case as being an invasive species, right? Disrupting the really previously potentially healthy ecology of the bladder, or of the bladder having a non-healthy ecology, allowing niche space for the E. coli to then invade, right? But in both cases, an invasive species dysbiosis issue. And another kind of figure that I've seen too that has driven this idea home for me is the meeting of the waters in the Amazon, where you have the Rio Negro here, this black water, and it joins here at the confluence, right? The Amazon River, which is this white water, and you can kind of see that the waters, merge but don't entirely mix until farther on. And how these rivers join, and the biota that's in it is really important, of course, to the economy and just health and conservation of this region. Now, between the two rivers, other than just their color, we have differences in temperature, flow speed, pH, dissolved sediment in the waters of the two river. This light colored water is rich in sediment from the Andes, whereas the black water running from the Colombian hills and interior jungle is nearly sediment free and is more colored by degrading leaf matter. And the microbial composition between these rivers is important too, like a developing UTI per se. And this is something that we really need to kind of keep an eye on the health of this, right? Like as our planet continues to change. And so just when we try to investigate this and we're trying to make models to really kind of understand infection, my group is always kind of looking for inspiration that we already see in nature because the answer or even the future prediction of what's going to happen could already be out there, right? But it's manifested in different form. And can we learn from those examples and apply them to biomedicine? And so with that, I am going to turn it over to Owen Hill, who's kind of asking a question. Can we use experimental evolution to learn more about the genetics underlying an invasive species in the bladder? Except of course, we're going to be doing this in vitro, as that's how our lab traditionally conducts our experimental evolution. But he is performing adaptive laboratory evolution to understand how E. coli evolve resistance to lactobacillus. So with that, I'd like to thank my collaborators, my wonderful lab members who really contributed to an intellectually diverse environment where we're thinking of anything from biomedicine questions like the urobiome. I have a student who is interested in E. coli and the environment and watersheds and the evolution of starvation. I have another student interested in bacterial epigenetics. I'd love to talk about that with other people at sometimes. Another student that's looking at population dynamics and mathematical models. So a really kind of rich kind of intellectual mixture that we have going on there. What that contributes to what my current and prior funding looks like. This work has been supported by Dannon, NIH, my R35 Mira, and the Army Research Office at some points in it. And again, just my incredible lab. And again, I'm sorry that I cannot take questions today, but honestly, this is a lot of Owen's work, so he will do fantastic at answering any of your questions and hopefully getting you all excited about the potential of experimental evolution. Thanks.